It is important to understand the role of the mitotic spindle. The mitotic spindle is made up of microtubules and they are anchored to the centrosome. The centrosome can also be termed an MTOC or microtubule organizing center. This forms the base for those microtubules to grow, extend, and eventually attach on to the chromatids. Keep in mind that when cells are not dividing or in G0 phase, they only have one centrosome present. Remember that this centrosome will duplicate in the S phase. This is to ensure that each daughter cell has one centrosome present, so that when it goes and enters into the cell cycle, it can then duplicate it as well to have the process go smoothly. Spindle fibers simply consist of the microtubules that attach onto the chromosomes. And remember, they're attaching on to the specific location at the kinetochores, which are present at the centromere. The centromere is simply a pinched off region of the chromatid where the sister chromatids are going to be held together. There are differences in this process between different eukaryotic type cells. Animal cells, as discussed, typically have a cleavage furrow that forms and that pinches off between the two cell membranes to eventually form the two daughter cells. Plant cells, however, have a cell wall. As a result, when they undergo cell division, they have to form a cell plate. This is made by vesicles from the Golgi apparatus that will line themselves up and form a small cell wall between the daughter cells. They will slowly start releasing cellulose deposits to form the cell wall. This will then grow, and eventually it will pinch off the two daughter cells. Let's take a look at this here and try to identify the different phases of the cell cycle. You can clearly see some of the cells have a nucleus. You can also see some of them, you can start to see condensed down chromosomes. In some of these cells, you can see these chromosomes moving towards the metaphase plate. Some will have a metaphase plate, and some will clearly have the chromosomes or chromatids being separated from each other. In some of these cells, you can see the appearance of a cell plate that will eventually form the two daughter cells. Keep in mind that this process of mitosis also occurs in prokaryotic cells. In this process for prokaryotes, we are going to produce two genetically identical offspring. This is done through the process of binary fission. Remember, in prokaryotic cells, you have one circular chromosome, and this chromosome is usually attached to the plasma membrane to anchor it. In the process of binary fission, you are going to get the DNA replicated. When the DNA becomes replicated, the copy of DNA will also become attached to the plasma membrane. However, it will become attached to the opposite end of the prokaryotic cell, which we can see in the diagram to the right. These cells will then undergo cell division. You will get the separation of the DNA, the formation of a cleavage furrow, and eventually the formation of two identical daughter cells. So you have the presence of a cleavage furrow that also occurs during cell division here, very similar to what you would see in eukaryotic cells. So remember, in prokaryotic cells, you would still have the G1 phase, you would still have S phase as well as G2, as well as mitosis. Overall, the cell cycle is a very complex process. During growth and development, there's going to be lots of cell division. In humans, this cell division occurs during embryogenesis, but it also occurs as tissues grow and also as tissues need to be replenished. In a mature animal, some cells can grow, but hardly ever divide, such as your nerve or muscle cells. This means that most of their time is going to be spent in the G0 phase. Some cells are always dividing, such as your skin cells, gut cells, and blood cells. This is because these cells need to be replenished at a very high rate. As a result, very little time is going to be spent in the G0 phase, and most of the time is going to be actively spent in the cell cycle. And some cells will only divide during certain times, such as in wound repair or tissue remodeling. Also, some tissues will grow and develop, say, on the onset of puberty. So as a result, they are going to be only activated into the cell cycle when the appropriate time is present. The cell cycle has to be very tightly controlled because any errors in the cell cycle will typically lead to the development of cancer. Cancer, by definition, is uncontrolled cell growth, either through continuous cell division or from the presence of immortal cells. These cells will not undergo what is termed apoptosis or cell death. They will simply reside and keep accumulating errors to eventually replicate themselves and spread. There's various types of checkpoints and also tumor suppressors that will help to control cell growth. These are very important in controlling the cell cycle to ensure that any cells that do not have the proper genetic information do not continue to divide and do not pass on mutations to their daughter cells. Factors that help to drive growth and division are called oncogenes. Oncogenes typically stimulate cell growth. There are also various factors that can prevent cell growth, and these are called tumor suppressors or tumor suppressor genes. The balance between oncogenes and tumor suppressor genes will help to control the cell cycle through the use of oncogenes to stimulate the cell cycle or through the use of tumor suppressor genes that will shut down the cell cycle. The cell cycle can also be controlled through the use of cyclins and their cyclin-dependent kinases. Remember, kinases are simply enzymes that phosphorylate other proteins. So cyclin-dependent kinases are simply kinases in the cell that will act to phosphorylate things. However, they're cyclin-dependent, which means that they have to be activated by the presence of their specific cyclin. A cyclin is simply a protein expressed during a certain time or phase of the cell cycle. By producing cyclins at specific times during the cell cycle, the cell can actually control the progression through the cell cycle. Through the use of these cyclins and cyclin-dependent kinases, the cell will be able to control the progression throughout the different phases. This diagram depicts the action of cyclin and its cyclin-dependent kinase. In this instance here, we have the cyclin-dependent kinase being produced throughout the different phases of the cell cycle. Note that CDK, or cyclin-dependent kinase, is present in G1 in S phase as well as G2. However, it is not going to become active until its respective cyclin is produced. We can see that the cyclin, specific cyclin for this cyclin-dependent kinase, is accumulating in late S phase and into G2. We can also see that there is a G2 checkpoint. This checkpoint exists to ensure that the cell has properly undergone DNA synthesis and also has grown enough in the two gap phases to ensure that it can carry out mitosis properly. As the cyclins accumulate in the cell, they are going to bind to their cyclin-dependent kinase to activate it. Once you get enough of these molecules, this is activated. This is going to push the cell through the checkpoint and allow for that cell to enter into the mitosis phase. This is going to lead to the production of the mitosis promoting factor. Once the cell enters into mitosis, the cyclin is going to be degraded. Once the cyclin is degraded, it will then leave the cyclin-dependent kinase. This will then remain in the cell. However, this will now be in the inactive form. So we can see that the production and control of the cell cycle is really through the production of these cyclins. The cyclin-dependent kinases are simply there waiting to become activated when the timing is right. The cell cycle is also controlled through the use of various checkpoints. We've already seen how the G2 checkpoint can be important, but there are many other checkpoints in the cell cycle. We have different checkpoints present in G1, G2, as well as the M phase. These checkpoints are going to be important in allowing for the cell to decide whether it is prepared enough to continue throughout the process. Various types of signals or ligands from outside of the cell can be used to either promote or inhibit the cell cycle. Remember, when a cell is not dividing, it is considered to be in the G0 or quiescent phase. It only enters into the G1 phase when the proper signal has been received by the cell. Once a cell receives the proper signal, it will then enter into the G1 checkpoint. This is a very important checkpoint for the cell cycle. It is the first checkpoint of the cell cycle. This can also be called the restriction point. This is literally the point of no return for cell division. This means that when a cell reaches this checkpoint, it can either progress through the cell cycle, but if it decides to do so, it cannot change its mind, which means that it has to undergo cell division. If it misses any of those processes or other checkpoints, it will then undergo what is called apoptosis or cell death. However, at this restriction point, the cell can decide to go back to G0 if it wants to. So this really is the point of no return. When a cell reaches G1, it can either decide to go back to G0 and not enter into the cell cycle. If it does decide to enter into the cell cycle, it must complete it fully or it ends up undergoing apoptosis or cell death. If the cell is damaged or the DNA is damaged, these checkpoints are very important in stopping the cell from continuing through the cell cycle. But once again, the consequence of not passing through a checkpoint after the G1 checkpoint would be cell death. This is a diagram depicting the various checkpoints within the cell cycle. We can see the G1 restriction checkpoint occurs within the G1 phase. We can also see the G2 checkpoint exists at the end of the G2 phase before mitosis. And we also have the M checkpoint. The M checkpoint is going to be important in ensuring that all of the DNA is separated out into the two daughter cells properly. So the balance between oncogenes and tumor suppressor genes, the balance between cyclins and cyclin-dependent kinases, and through the use of these checkpoints, we can see that the cell cycle is a highly controlled process.